A todas y a todos, gracias por asistir y venir a esta charla de un invitado que me da mucho gusto que haya tenido el interés en compartir con los centros de la UNAM, con nosotros, lo que están haciendo en un centro de investigación que a veces muchos no conocen, pero que es un centro de investigación de excelencia a nivel internacional. Tiene varios, bueno, él ya va a presentar todo, pero pues a aquellos que se dedican a las ciencias vegetales y que piensan que nada más uno puede dedicarse a ciencia básica, pues vean que hay a veces otras opciones y sobre todo que están en México, ¿no? O sea que a veces no es necesario salir del país, ¿no? Entonces, thank you so much, Anindya, to accept the invitation, and thank you, everyone. So go ahead and.
Thank you, Jesus, for nice introduction, and thank you all for coming. So I work in International Maize and Wheat Improvement Center. So we are known for maize and wheat, but we are also working in the area of dryland crops, which is our new crop last 5 to 7 years. Now, okay, so wheat, maize, I mentioned. Dryland crops, we work on sorghum, millet, groundnut, pigeon pea, and chickpea. Other than that, we keep genetic resources for mostly maize and wheat. And we have a sustainable agri-food system, it's SAS, mostly made up of agronomists, economists, social scientists, etc. And most of the SAS people are in Africa, and few are in Mexico City. So my laboratory or headquarters is in Texcoco, which is 45 minutes east from Mexico City. And we are present in many countries across the globe. So these are the accessions, number of accessions of wheat we have in our center. And these are the accession of maize we have in our center. So we are we keep all the germplasm of maize and wheat, and then we give it for free across the globe. We also develop new breeding products, which we also again share through our website, through international treaties. And one copy of all this seed goes to Norway, Svalbard. It's below, the main part of this gene bank is below ice. So if something happens, nuclear war or something, then human being can take seed from there and use it. So this bunker is nuclear war resistant and etc. etc. So this is the ambassador of Mexico in Norway. He basically, whenever we go and give seeds to this vault, the ambassador comes and we work very closely with Mexican government related to germplasm and etc. So CIMMYT, this is our headquarters, and we have centers across the globe. The biggest center is in, after headquarters, it's Kenya, and one in India, Delhi. So these three are main research station. Apart from that, we have fields and office in 80 countries, so across the globe. So most of the UN, United Nations members are part of it. And biotech research happens here, but unfortunately, we cannot bring it to the field, in Mexico, because of there is no regulation for gene editing. So we send all of our gene-edited seed to different part of the globe, in Latin America, in Brazil, Argentina, also in many countries in Africa. Now India accepted it as a non-GMO, so we also send to India for field trial, etc. But unfortunately, we cannot do field trial in Mexico. So why I wanted to tell this, I gave talk in other places as well, and I realized that in Mexico, very few people know about CIMMYT, or very few people know about CGIER, so which is very surprising to me. So I thought it would be a good idea to talk a little bit about what we are. And so as I mentioned, maize, wheat, genetic resource, sustainable agri-food system, and dryland crop. And 50% of maize and wheat in low and middle income countries is based on CIMMYT varieties. So parents are from us. And then you do breeding and you develop your product. And we also try to keep their product one copy in our gene bank. So anything is needed from there, you can get. So we come under a semi-UN or UN-related organization called CGIER. There are 13 institutes across the globe based on the crop. So we are CIMMYT. And then there is IRI, International Rice Research Institute, CIAT in Colombia, International Potato Center in Peru, then four research institutes in Africa, some in the Middle East, and CGIER headquarters is in Montpellier, France. And we have only one institute in US, in DC. It's called International Food Policy Research Institute, IFRI, but that's mostly for economists and social scientists. So science research is not their mandate. So these are all international organizations under CGIER, and CIMMYT is one of that. So here are all the names. So now let me come to my topic, gene editing. We are doing gene editing. So we have breeding, agronomy, agriculture engineering, and many other departments. Gene editing is one of that. So we come under Genetic Resource Program, GRP program, and these are the new building we are having. This is only for gene editing, funded by Carlos Slim Helú. So, so he donated money for this building, and we made a confined gene editing center there. So these are the crop we do, as I mentioned. So sorghum, groundnut, pearl millet, wheat, maize, and also PGNP we have started. These are our collaborators. Multiple collaborators are there. And these are our funding agencies. A big funding comes from Gates Foundation, FCDO, Australian government aid, Saudi government and European Union. So we get these fundings for our research, and then we develop product, and we give that product for free to global South. So we are non-profit. So CRISPR technology, as you know, these are very IP protected technology, and it's difficult, it's costly to get for making product and give to the farmers. But we are lucky to get Cas9 license, end-to-end license from Broad Institute and Corteva. They are now having pool of all Cas9 together. So they gave us for free. We can do it through our collaborators. Also, we can give it to our collaborators. But if it goes through for-profit, sometimes in Africa, there are some countries where government is not very stable. Always there is change and there is a problem. Then we give it to the local small companies, our seed. It could be breeding seed, it could be gene-edited seed, whatever. And they sell it to the farmers in small price. So in that case, definitely they will take some royalty from them. But if we go through government and we disseminate the seed to the farmers, then it's free. So that, and also we have taken Shark from Pairwise, end-to-end license. And recently we are getting Cas12a and Prime editors from Broad Institute. And every year, as we work with Corteva, Pairwise, and other private companies, every year we pass this audit excellence through stewardship. Private sector needs this. Otherwise, they will not allow you to develop product using their technology. So this, we every year, every 3 years, we pass this test, and we are certified. So how do we see gene editing? So if I divide plant breeding in two major areas, one is phenotype to gene, and one is genomic selection to your product. And the third would be your GM crop development. It's completely a gene to phenotype, a different aspect. So we see gene editing is this red line, where we can expedite breeding, where we can help breeding by doing rapid functional validation. We can create allele diversity. We can help in optimize allele in product line. And then by editing, we can do one-step trait integration. So we can really break down, or we can reduce the time for breeding. Now, this is a very favorite equation of the plant breeders. If I forget everything, if you change, this is genetic gain equation, if you change this breeding cycle, time for breeding cycle in half, your genetic gain doubles. So you reduce the breeding cycle in half, your genetic gain doubles. So by CRISPR, throughout this equation, there are application of CRISPR and benefit of CRISPR, but editing can really reduce breeding cycle in very like smaller time, and accordingly we can get more genetic gain. This is from Corteva. I took their permission to show this one. They are our collaborator. So a transgenic, a GM, it takes 13 to 15 years for deployment for them. Breeding lines, it takes 9 years because of the continuous backcrossing and cleaning up your product. But gene editing, they developed this waxy corn line. It took 4 years, 42 months. 2015 they started, 2018 product in the market. So it's much faster. So I requested them about this slide, they showed us. So, and country-wise, if you see, many new countries are accepting gene editing as non-GMO. So apart from Peru was red, but very recently Peru has gene editing regulation, and it's case by case. You can do it. Mexico is red, unfortunately. And most of the Latin America, South America is green. Many African countries are green. Now China, India, Australia, Indonesia, these area is green. I kept it yellow, but NGT1 in Europe now accepted. It's coming out as a law. Now it's needed to be seen how many countries follow that and what they do. I believe countries will get their own regulation to work as well, apart from this one. So it's pretty interesting and pretty positive. It's not taking the track of GM, rather its track is separate and better, I would say. Now coming to the technology, what we use in CIMMYT, these are all technologies we have and established in our organization, but we have license for research, end-to-end product development license from Cas9 and Sharp. And now we are trying prime editors, and we are also trying Cas12a. So these two we will get very soon from Broad. Then we have a toolbox ready to do the research. Now, one example, I wanted to do one thing. I wanted to give a glimpse of the whole thing, not one particular project. So what we do, so that intention is to have collaborations with you guys, get student, exchange student, something like that. So this is a devastating disease in Eastern Africa, maize lethal necrosis. It caused by two viruses, MCMV, maize chlorotic motile virus, and SCMV, it's a potty virus. When they come together, they create havoc. And eastern part of Africa, they lost a big amount of crop in 2012, 2013, that time. And it was spreading throughout the Africa. So these governments came to us and told us to develop something. So CIMMYT started checking their gene bank for any natural resistance. And then we could find one line, which is a Thai line. It's from Kasetsart University. They developed that line that has a QTL, which is resistant to these viruses. What we did, we started mapping that QTL, and then finally we narrowed down to 150 genes. Then we knocked out all 150 genes one by one in collaboration with Corteva, and we could come to a gene which is basically a peptidase. So it's an ACE gene. We could find an ACE factor, and once we knocked out that ACE factor, this is the result. So we planted our edited lines where we knocked out one ACE factor peptidase, and this is called Naivasha, Kenya, where it is MLN hotspot. These viruses are everywhere. And this is our edited lines. And these are all commercial varieties of that time. Different companies are selling maize. They are becoming like this, but the edited one started showing different kind of. While doing this, I'm not going in detail of each of the science part. While doing this, we could find another one new set of genes. There are dwarf maize, small size maize. Their life cycle is very short, like 2 months, they are seed to seed. So we use that dwarf maize for research purpose, for faster research. But we found that dwarf maize, if we edit EIF4, elongation factor, everybody knows this is a very common viral resistance S factor. The small maize is resistant to these joint combined virus. But if we edit the same gene in tall maize, the normal maize, they are not resistant. Then it was a very funny, suddenly while doing research, we found that, and then gradually we checked there is a natural deletion in the 4th exon of these small maize. So if we delete EIF4E1 and natural deletion, when they come together, they are resistant. So it's a new finding, and what we did copied the same in the normal maize, and again it's resistant. So now what we are doing, the peptidase which we identified in our QTL mapping, and also this EIF4, we are bringing them together, developing the hybrid, And by 2027, we will release this hybrid in Kenya. So, and this will be the first product, gene-edited product in Africa from a non-profit organization. So we are pretty excited. And these are our collaborators funded by Bill and Melinda Gates Foundation. Initial funding, little bit USDA gave. Then Kalro is the Kenyan organization. We did major research along with Cortiva, and this is the AMSAC of Kenya. This is TAC. So it's a joint conglomeration of companies. So we all work together and we could generate these projects. So this is our most advanced gene-edited product in field. And we are now making the hybrids. Now we'll give the hybrids to the farmers. Seed. We are also working in strigolactone pathways. Not only pathways, which weed, you know, the striga is very common and very problematic for maize, pearl millet, sorghum, and many other crops actually. So we are working with Harro Bomister from University of Amsterdam. He had a science publication last year where he showed this pathway, and we knocked out 7 genes. And those 7 genes, we have separate line. Now we are sending seed to him, and he will then do the biochemistry, and he will see. And then our interest is to, he has a collaborator in Switzerland. There, field trial will be done. So this is the plan. So this is another one project. Mohan is our scientist. He is working here. We also take students. So Simran Anne did her PhD in Iowa State. We basically funded her. So half research our place, half research Iowa. Simran is doing in Mexico City the whole research, but degree will be awarded from India. And recently, Nicole came from Guanajuato, and she was there for a year, and she is now trained in maize editing. So we do baby boom ushal mediated editing or transformation, which is patented by Cortiva. So you cannot make product, but that's faster. That's very good. We have also established non-baby boom ushal system for that. One more project in maize we are having. It's a new tool, 3A, basically RNA targeting CRISPR in type 1. So we are using with Blake Weidenheim. Blake was the famous postdoc of Jennifer Doudna in Berkeley. And Blake is now working on bringing up new RNA targeting tools for viruses. So we are collaborating with him and also with Markita. So we have 3A, we have developed, and we'll publish it very soon, that 3A can be used in plant as well. So, and we have targeted that same MLN virus, and we can show that by 3A we can chop RNAs, because both are RNA viruses. One is negative strand, one is positive strand, and we can reduce the virus load in a significant level by this tool. So this is a new tool. So little bit of tool development we do, because we are not allowed to do basic science. We are not allowed. We are allowed to develop product and give to the farmers. So we collaborate with many basic science labs for the tool and technologies. Okay, I'm not going in detail. In wheat, Raphael joined us very recently as an associate scientist. Easter came from Missouri. Josh came from John Innes Center. They are working on wheat. We are working on multiple wheat diseases. And one example I will show, we all know about wheat rust. Rust is there forever. There are many breeding lines. People try it next year. The resistant barrier is gone. Another one new rust strain comes and again devastate. Again, people go for breeding and make a new line. So rust resistance in D, there are 3 genome hexaploid wheat. So D genome, there is one natural variation, one SNP in STP13, sugar transporter, that basically gives rust resistance. But most of the elite lines, wheat lines, they don't have this SNP. prime editing bit. Added a few new things like prime editor has, it's called a PEG RNA. It's not gRNA, it's called PEG RNA. So the exposed part of PEG RNA basically get chewed by other enzymes, etc. So giving a knot there, pseudo knot, and this kind of stuff we have done a little bit. And now we did prime editing of that. Just no cutting, no double strand break, just changing the SNP. So we could do that. These are our sequence lines. And then Raphael did all this visual scoring of RAS disease. And then you can see there are changes in RAS efficacy in the prime edited line. And some of these peer genes and sodium oxide dismutase, these genes goes up during the infection. When there is resistant allele present, then it goes down after 24 hours. So same pattern we could see in our prime edited line. So we just changed one SNP. Now we have submitted in one of the journals. It will come out soon. So we changed A genome, B genome, D genome. So we have variants A, B, D, all three prime edited, A prime edited, B prime edited, AB prime edited. So different variants and different kind of doses of this SNP is giving varied result. So this is another one work we are doing. Some fungal RNA, fungal DNA and biomass measurement that also we have done. If you see, the biomass is very high in case of non-edited line. In different edited line, different kind of lower value of biomass we have seen. So this is another one work. I'm not going in detail. We are also working on Fusarium head blight. So what we are doing cisgenic. So in this project, we are sequencing Borolog 100, which is parent of many wheat lines, one of the parents. So we are sequencing it. We are basically doing RNA-seq in the spike as well as leaf. Then we are finding out spike-specific promoter. And by knock-in technologies, we are putting those spike-specific promoters in front of our genes, resistant genes, so that those genes get highly expressed in the spike. So that is also another one work. We got funding recently, and we have started this work. A very recent thing on pearl millet we have got. So pearl millet is a very robust crop, and it's very popular in Africa, India, Pakistan, Bangladesh, all this area. Now, there was no report of pearl millet transformation and editing because it's very hard to transform. And we got a project from Gates Foundation where we tried to, and pearl millet flour, when you mill the pearl millet, you get flour, but you have to eat it immediately. Otherwise, it has a lot of lipids, and they make foul smell after like 3-4 hours. You cannot keep it or store it, so that farmers cannot ship it. So there are issues. So we wanted to make pearl millet, it's called rancidity trait, so rancidity free. And for that, we have identified 2 pathways, one oxidative, one enzymatic rancidities. So both the pathways, lipase genes, lipoxygenase genes, we are knocking out. And we are also playing little bit with the PUFA, like polyunsaturated fatty acid. We are reducing that to monounsaturated. So, and we have taken all the African lines, which are popular in Africa. We have established transformation, amplified those genes. It's a 63 days transformation protocol we have established. And then these are the edited lines. You can see this is a white stripe. This is basically a heterozygous PDS knockout initially we did. So you get white and green stripe. And then these are basically edited lines for 4 genes we have identified in the rancidity pathway to get rid of. And in November, it came as a plant physiology cover. We haven't given data for our editing and rancidity. We just published transformation and gene editing in Pearl millet. So this is the first report. And now we have all the 4 lines ready. So seeds are getting ready, like we are taking them to next generation so that they are homozygous. And we have started doing fatty acid analysis. So our major interest is to see, we have knocked out all 4 genes, our target genes in pearl millet. Now we are expecting fatty acid profile will change, and this will be helpful against rancidity. So that is another work we have done. And also we are crossing these lines as well as we are doing multiplexing. So nowadays we frequently do multiplexing for 6 to 8 targets. And for all of the genes, we do 2 targets actually, exon 1, exon 2, early exons when we need to knock out completely. So multiplexing is happening pretty well, and all 4 we are doing multiplexing. We have a project on PGNP. It's funded by USAID. Then Donald Trump came, USAID is vanished. So we don't have any more money, but we have done few things in collaboration with Yiping from University of Maryland, who makes different kind of gene editing tool. He's very famous now. And he is establishing 2 things. A new barrel came for Biolistic. It's called flow-guided barrel, where you can target bombardment and the copy number is 2 or 3, just like agrobacterium. So whether it happens for all kind of crop or not, we are trying to see that with Yiping. He has published in Nature Plant 2 months back, I guess. So that's a new tool came that we are using. And also we are doing ACM-mediated transformation so that we can avoid the tissue culture part directly in the, and that's another one. Bayer is our partner, and these 2 our people. And we have established transformation of PGNP. Although it's lengthy, it takes 6 months, because seed to seed is almost 6 months, 7 months like that. And it took us some time to establish PGNP in our greenhouse in Mexico, because they usually grow in a very hot and airy temperature. But these are very popular legume again from Latin America, from Africa and Asia. So to test this, the project was on cry genes. So cry1 it's GM, it's not gene editing. So we have developed this GM. Now we are supposed to send this to Nigeria and Burkina Faso, targeted for those 2 countries only. But now no money, so we cannot do anything. So we are waiting. We are waiting for change of the government. But anyway, so I'm not going in detail. We are also working on groundnut. It's aflatoxin resistant groundnut. First phase is over. Now we are getting the second phase from Gates Foundation, from Texas A&M, Venu is partner, and also Tropic Bioscience from UK, and 2 Blades. These are the 2 companies are involved in this project. So we are targeting 2 different ways. We have established transformation for groundnut. Again, regeneration takes long time. We're trying to narrowing it down, and also for groundnut, we can do JL24. It's an African elite, but it's elite of 10 years back. There are new elites, which are very difficult to transform. So we are trying to establish new elites as well. And then in this project, we are trying both ways. Green plant has genes against aflatoxin, but aflatoxin is more in the after harvesting. So when you store peanuts, their aflatoxin infection becomes very huge that time. during the green plant, when the plant is in field, it's not that much. So there are two schools of thoughts. Some scientists think that green plant genes cannot help S genes. Some scientists think no, green plant can help. So we are doing both. So seed coat mediated resistant we are doing with Texas A&M. So seed coat has some chemicals which gives natural resistance. So we are trying to increase those chemicals. So that is one. And second thing, we have identified few genes for the green plant. So that's also we are targeting, knocking them off to see what happens. So it's in early stage. First phase, transformation establishment, editing establishment is over. But the next thing is that. So this is our team. Raphael joined recently. He probably came here to give a talk. He worked quite a while on nitrogen use efficiency. We are having a new project funded by Bayer, which is BNI wheat, biological nitrogen inhibition. So in Japan, they crossed wheat with wild grass. Half of one chromosome came from wild grass to wheat, and those wheat plants are NUV efficient, quite significant NUV efficient. Now this is 300 MB fragment. So we got a funding to sequence that 300 MB, chop it by CRISPR, make different small versions, and make different line, check nitrogen use efficiency, BNI efficiency. It's biological nitrogen inhibition. So, so that work we'll be doing, and we are looking for collaborators. That's one of the reasons I am here. I am taking some rhizobium and other bacteriums from okay. So that's one of the reasons. Easter came from Missouri. Mohan came from India. Danny is our consultant, came from John Innes. Han came from Pearl Millet Center in Beijing. These are our students. And I forgot to put actually 3 more people. So these are our local staffs. We have 2 types of recruitment. One is called internationally recruited staff, one local internationally recruited scientist IRS, and locally recruited scientist LRS. So IRSs are under the rule of UN, LRSs are under the rule of Mexico government. So these are all our expert transformation people. They all do transformation, admin stuff, and our greenhouse stuff. So this is it. Our collaborators, funding agencies for gene editing. Some of the, I put this photograph last night to make it look nice so that I get some students. Okay, so, and thank you. Any questions?
I think we have time for questions. So is there any question, Esperanza?
When you get reduction of aflatoxins, do you avoid infection by Aspergillus?
We are not in that stage yet. As I mentioned, it's a new project. First stage, we have established transformation, editing, etc. Now in the second stage, we will do all these tests. But our Texas A&M collaborator, they have found out some proteins in seed coat which has natural ability to work against aflatoxin. So those are published. So that part, the aflatoxin infection reduces significantly if we use those chemicals. So they purified those chemicals and tested in lab, but not in field yet. So now our interest is to improve production by GM and also by gene editing. Gene editing, we are doing promoter modification to make it more in the seed coat. and we are expecting significant reduction of aflatoxin. So we are in early stage, so I cannot tell you what exactly happening.
Thank you.
Thank you for your talk. How stable is the resistance that you get for the viruses?
Yes, so viruses, that's why what we have, so peptidase edited once, it came out two years back. So these 2 years, it's holding, and we believe it will hold for long, but we are adding EIF4 on top of that.
Okay.
And we have found 3 more QTLs, which are in chromosome 3, one is chromosome 10, like that. So we will find map those always. It's like antibiotic, you go on adding molecules.
Exactly.
But it's still there, 2 years it's still there, and we believe it will not go so easily. But we are also getting ready with the second line of product, third line of product, like that, with new targets.
Yeah, good idea. And this resistance is effective in different backgrounds, genetic backgrounds?
Yes.
It's effective in different.
Yeah, so we use white corn. In Africa, as you know, they only eat white corn.
Yeah.
Yellow corn they give to the animals. So that's a challenge. So we have very good tropical corn, but we cannot use them in Africa because they are yellow.
Yeah.
So whatever white lines we have in Africa, elite lines, we used it. Like 7 or 8 new hybrids, and they are working well. So we are using it.
Yeah.
So in this context, we have a project, we are taking good tropical lines, drought resistant, and we are changing yellow to white by gene editing. And we will, for Africa.
Good.
¿Alguna otra pregunta? No. Okay.
Hey, thank you. Very nice talk. So this is out of ignorance. Why is so big the difference between transgenic breeding and traditional breeding? Why? I mean, it's very comparable in time, in terms of time, right? So why is that transgenic?
Transgenic takes long time for regulatory approval, and it's very costly. It's very costly, and regulatory approval at least 6 to 7 years to 8 years. Okay. So it takes long time. It should not, but regulation is like that. So.
And regarding genome editing, these 4 years are also considering testing the plants in the field in terms of yield?
It's diverse, I would say. Like for MLN, we know the gene, and we can immediately knock out the gene in edit line. One of the parents, if it is recessive, then both parents you should edit. Otherwise, one of the parents edit. So within 6 months, you can have 2 generations, edited lines. And you can test, do field trial, even faster than 4 years you can do. But if your gene discovery part is involved, then some countries require 2 years data, field data. So it depends, it varies from country to country. But theoretically, within 2 years, you can get a fixed edited line, like homozygous edited line, right? And after that, it depends on some country, 2 rounds of trial, some country, 1 round of trial, it varies.
Okay, thank you.
I think Mashal.
Hi, thank you so much for the great talk. So something I really like that you showed is, I think it's great to have more exchange among the global South, and I'm a big proponent of that myself. So like you showed Indian students coming to Mexico and vice versa. So just what are some routes that you found that have been like fruitful in that kind of thing, and what have been some of the major challenges?
To disseminate the seed in global South?
To have more exchange of ideas and scientists within the global South, like for example, between India and Mexico and so on, Whereas we've been coming from a time where it's been very like global South, global North collaborations.
So big problem is funding. So China, India used to put a lot of money in CGI here. They have stopped it. They think they tell us that we are equipped enough. We don't need your help. That's one government level problem. Still, India has some research collaboration with CGIAR. So CGIAR started in 1960s, and then Green Revolution happened from CGIAR. By the way, CIMMYT is developed by Norman Borlaug, who is known as the father of Green Revolution, got Nobel Peace Prize and all these things. So that time, whole Asia, Africa, Latin America, they were happy to get CGIAR and collaborate with CGIAR, get the seeds and all new technologies, etc. But gradually, few countries, they have developed their own research. So they are not very keen to collaborate anymore. So that is one of the problems for CGIAR. Second thing, we see that each country has different rules and regulations, target traits. So it becomes very difficult for us, like 5 countries in Africa, 5 different traits for maize, right? So always we cannot do that. So that is another one problem. And third, there are political unrest, because end of the day, our main target is to give seed through government for free. But due to political unrest and all these things, we cannot do that because we don't have any permission or mechanism to give seed to the farmers. We can develop the seed, we can give to the local organizations, like government agriculture organizations. They multiply the seed and then they disseminate. So sometimes government is not ready to do, or political unrest or something, then we give it to the small companies. So these kind of problems are there. And also technologically, many countries has many different regulations in Africa, Latin America. And GMO was a nightmare, but gene editing at least getting traction from many countries. So gene edited product now we can gradually give to, but if there is natural variation by breeding, if you can do a product, we don't do that. We means gene editing team, we don't do that. If natural variation is there, breeding is possible, then let them do it. We only target where it is difficult by breeding or it is very slow by breeding, like that. So this kind of thing, some countries still, although it is approved in paper, but when you go actually, there are a lot of problems in dissemination. It doesn't happen.
Yes, Esperanza.
Are you evaluating life variability of seeds in the Norwegian bunker? Because you're very happy to have them. Yes, they test regularly.
Every gene bank has certain rules and they come under crop trust, all the gene banks. And there is a regulation that every 3 years or 5 years, you have to regenerate, check certain marker, run some markers, check few things like that. So Norwegian bank, yes.
You do that?
Yes, we do that.
Okay.
Now, crop-wise, it is different. Some crop 8 years, some crop 10 years, some crop 5 years. Depends on the viability of the seed. Yes.
Yeah.
The problem is it requires a lot of funding, like in millions, right? Some of like rice and all this thing, and putting them all in field, checking viability is a lot of work and a lot of. It's not technically not difficult, but funding-wise, logistically very difficult.
Arnaud?
Thank you Thank you for your talk. I have a question. If CIMMYT has the goal to keep the germplasm, and most of the new technology require information of genome, do the CIMMYT has a goal to keep information of each genome for each variety, or?
Yes, so not everything. It depends on, again, which is unfortunate, but it happens. CIMMYT scientists apply for whole genome sequencing of maybe 10 different crops, 10 line each. But funding agency is telling only do maize, rest you don't do. Then we have only maize data. So something like that, it happens. But almost 3,000 resequencing has been done. So each center has genome sequencing projects, and those sequence data, marker data, all are available with the team. But I must say, it's not like all lines what you have. That's not possible because it's costly, right? So a scientist comes, he or she is interested in certain area, in certain line, we sequence those. But there are centralized effort from CGIAR to sequence most important elite lines. So in rice, I know they have sequenced almost 7,000 accessions. Maize also, they have sequenced quite a bit. But wheat and others are not that many. So it varies. But we keep those. Whatever we do, we keep those, and it's in Dataverse. So you can access it freely.
Is there another question? No? Well, then I just have a very quick, short question. You mentioned about this crop that was finally could be transformed. Have you tried with many other crops and you didn't succeed it? Or how difficult was to succeed this one?
Which crop you were mentioning?
One of the last ones. It's just that I don't.
Groundnut.
No,
Pigeon pea.
No, it was about your plant physiology publication.
Oh, okay. Pearl millet. Pearl millet. Millet.
Yep, that one. So
Yes.
how difficult was to finally transform this crop?
Yeah. So for these crops, millets, it was not a common practice of using growth regulators. So like baby boom ushel, WOX5, there are others also. So in pearl millet, we had to use growth regulators to make it regenerate. So that's new finding we did. Previous efforts, whatever people tried the normal way, it didn't work. Or even if it worked, it's very poor performance. But when we added growth regulator, we could regenerate many.
Okay.
So yeah, it took us like 2 years.
Yeah.
Like 3-4 people continuously worked 2 years with many failures.
I can see. Well, thank you so much, Anindya, and I think that's all. Thank you for coming.
Thank you. Thank you.